with Professor Ricardo Della Favera, the recipient of this year's ISNAF Lifetime Achievement Award. Congratulations and thank you for this interview.
Thank you very much. Thank you. I'm honored by the award. Yeah.
Professor Della Favera is Professor of Pathology and Cell Biology and Director of the Institute for Cancer Genetics at Columbia University. Professor, you are a recipient of numerous prestigious awards and are a member of both the National Academies of Science and Medicine. And you are one of the world's leading cancer geneticists and lymphoma researchers. So we'd love to hear from you a summary of your research.
Okay, maybe to understand my research, I'll do a brief historical excursus in my activity. So my research, I was trained as an oncologist hematology at the University of Milan, and I have always been interested in research, in particular in cancer that affect blood cells, so leukemias and lymphomas. I was fortunate to initiate my research activity at the historical time of cancer research and cancer biology research in general, when we could finally identify and clone genes. So cancer has been suspected for a long time, for even more than a century, to be a genetic disease, a disease that starts from alteration in our genome, in our DNA. But we didn't know what were the components of the genome, which were the genes, the actual effectors that determine the cell destiny and fate. So in the late 70s, finally cloning, recombinant DNA technology was developed. And my original research, when I moved to NIH as a postdoc and then as an associate, was to clone so-called human oncogenes, genes in the human genome that have a normal function, but clearly were altered in many cancer types. So the first human oncogenes were cloned, and then the attention of the research turned to whether they were altered in which cancer types. And the other turning point were some of these chromosomal translocations, events, specific changes in chromosome that were visible at the microscope and specific for certain leukemias and lymphomas, were shown to actually affect directly breakage or changes of these genes. So finally, we had targets in DNA, changes in DNA that were clearly associated with different cancer types, particularly leukemias and lymphomas. This was important because for every disease, the best way to treat is to actually understand how they develop. And as you all know, still now, but less and less every day, cancer has been treated in a very specific way with chemotherapy, which are toxic to every cell, to tumor cells as well to normal cells. And when in different cancer types, altered genes were identified, we had a handle to directly target abnormal events in the cells, which are different in different cancer types. In particular, my research focused on lymphomas, which are the fifth most common type of tumors. And with another major technological advance, we're now talking about the 20 years ago, which is DNA sequencing. We could sequence the genome of normal cells and tumor cells and compare them. And instead of having single genes altered, we had the whole cadre of all the genes altered in any particular cancer type. And there were two major conclusions. First, cancer is not one disease, it's probably more than 200 different diseases that differentiate for the type of genes that are altered. And so the lymphoma, we now know around 100 genes that in different cases are altered and represent their protein product. Genes makes protein, the targets for attacking therapy. And so in particular, the research of my group moves from identify the genetics into reproducing those alteration in mouse models. So we show in mice that if you have a certain gene change, you cause lymphomas. And we understand the biological function of these genes. And sometimes there is a drug that we can test in mice. Sometimes we collaborate with industry or with clinicians to develop or test new drugs. This is in a nutshell where we are in lymphoma and where I think the whole field is. If I have to summarize, I would say we are coming out of 30 years of understanding how cancer develops, and we are entering, probably we already entered, the era of targeting those lesions, of therapeutic progress. And there are already some examples of cancer that have turned from lethal to at least a chronic disease is not cured.
What do you think are the most promising future directions in cancer research?
Well, it's as I just said, we now know a lot about how cancer develops, and we have to painfully develop drugs, or major development has been immunotherapy that attacks specifically cancer cells, leaving alone normal cells. So increased specificity and much decreased toxicity. You're going to see in the next 20 years that more and more cancer will be, as I said, turned into from lethal to chronic diseases that can be managed and some cured. Unfortunately, a number of cancer remain quite bad, but progress is there. The pathway is open to develop drugs, more and more drugs that are specific for even those lethal cancer types.
Is artificial intelligence having an impact on cancer research?
I think this is probably the beginning of a new era. We are often facing an enormous amount of data, the complexity of which is very difficult to dissect and understand. So artificial intelligence can capture all the available literature, all available data, assemble it in a way that even most sophisticated algorithm can do. So I think the impact will be enormous. In diagnostic, there is a lot of discussion whether artificial intelligence will substitute the diagnostic by humans. They will be has to be supervised by professionals, but certainly the capability to handle an enormous amount of data together and to interpret and organize is amazing.
ISNAF aims to bridge scientific research between Italy and North America. How do you see yourself in having a role in building this bridge?
Well, first of all, ISNAF doing a great job because there is a large number of young Italian scientists that every year migrate to the United States, enter different laboratories, and are facing a new professional environment. My role is to participate into ISNAF various initiatives, the very important awards they give out. So I supervise an award that is specific for hematologic malignancies. And also a new initiative has been the mentoring of selected Italian students that are considering what is their future, where they're going, where is their career is going, what they should do. And I think a number of ISNAF associates is talking, advising, guiding very talented Italian students that are exploring the way to come to the US and what they should seek in the US.
What can you tell us about your current professional ties with Italy?
Well, traditionally, I always maintained through the years strong collaboration with the Italian scientists in my field. I also, through the years, I think, trained as a postdoctoral fellow, as a student probably more than 30 Italian young scientists that some of them now occupy leadership position in Italy. So my connection in Italy has always been strong, and it is still strong.
As always, let's conclude with a piece of advice for our young members.
So the young members, well, the advice is enter the research as a career only if that's your passion. You really have to think that's the only thing you want to do, because it's difficult, but very exciting, highly rewarding life. If you have that passion, it's a life not free of sacrifices in other areas, but you get addicted to it. And if that is where your heart brings you or your brain brings you, that's what you have to do. And the second advice is, in research, always identify, select a major goal, because even if you don't get there, you have a feeling that you are working for something very important. So be ambitious.
Is there anything else that we haven't covered that you'd like to share?
No, just thanks to ISNAF for all its activities, and of course for this award, which is an honor. I look at the list of my predecessor in this award. Is extremely it's an outstanding series of outstanding people, and so I'm very honored and very grateful.
Well, grazie mille, Professor Dalla Favre, and congratulations again.